All right. 3, 2, 1. Why, hello there, guys. Welcome back again to the Evidence-Based Excellence podcast with your host, Mr. George Cresswell. Going back into something really, really important today. First, I just want to say hello to you. I want to say hello to you. Hopefully, it doesn't lose focus. I've got the secondary camera set up at the distance this time rather than being right in front of me. I think it was something to do with this arm, this microphone arm being in the foreground, that was screwing with the focus a little bit, so to speak, and my phone can actually focus better with all of this different stuff going on. So hello to you, and hopefully we don't lose anything. Hopefully we don't, end up with any bad footage. And yeah, hopefully it just all goes according to plan. I'd be happy if the other camera stays in focus this time, because then you get to see my lovely doggo. You get to see Bailey. Say hello to Bailey, everyone. She sits up there in the corner. My little doggy pillow or doggy cushion. That is our black Labrador Bailey, and I don't know if you can hear her on the microphone, but she's actually right here also. So I have cushion Bailey and I have actual black Labrador Bailey with me right now, chilling in the room with me because it's hot as balls here currently. And we have a nice little tower fan going in the background that you may or may not be able to hear. I don't know. But she heard her name, and now she's wondering what the hell is going on. I was looking at me like I have 3 heads. So, yeah, we're going to get on with today's episode, which is all about protein, the fundamentals of the macronutrient protein. Obviously, it's good to have a good knowledge of protein, whether it's for your next pub quiz, or whether it's because you do, in fact, have an interest in health and fitness and want to know more about the nutrient of protein that's going in your body and how it actually pertains to your health and fitness goals and all stuff like that. This may be a multi-section podcast. I don't know. We'll see how we go. We're going to be going into a little bit of the background science with regards to protein, how it's formed, what forms it, all stuff like that. And then a little bit about its, utility and intakes and all stuff like that a little bit later on. So this will go hand in hand with, I think I wrote an article about protein on my website. Can't remember if I did. We'll see. But going into it straight away with the question, what is protein, or what are proteins? I'm going to read you it straight off my notes that I have made for this episode here. And the big thing you need to remember is proteins are fundamental structural and functional elements within virtually every cell of the body and undergo extensive metabolic interactions within physiological processes, predominantly regarding the metabolism of energy and other nutrients. And obviously it also goes into the repair and growth of tissues also. That's one of the main processes that dietary protein is involved in. Proteins are made up of extremely small constituent molecules known as amino acids. If I turn this into an article, which I probably will for my website, you'll see that abbreviated as AA or AAs, like AAS, amino acids. When they're combined into long chains known as polypeptides, another word for protein, these form proteins and hormones that have distinct functions within the body. These polypeptide chains and amino acid sequences are generated from sequences of nucleotides contained within our genetic code as humans, contained within our GNA. Well done, Josh. Well done. Now, contained within our DNA. Okay, there's 4 in total of these nucleotides, and they're known as guanine, thymine, adenine, and cytosine, okay? And they are arranged together in two forms known as base pairs, and that is adenine to thymine and guanine to cytosine, okay? A to T, G to C. Amino acids and amino acid sequences are made from processes involving sequences of these base pairs, okay? So ultimately, amino acid synthesis comes from our DNA, okay? It comes from our genetic code. Some of them as well, obviously we'll get into different types of protein and amino acid classifications later on when we talk about those that have to be taken in dietarily and those that can be synthesized and formed via other means endogenously within the body. We'll go through all that a little bit later on in this episode. Proteins are abundant within the body in various forms and tissues. That's another thing you need to know as well. Let's take the example of an average, just say an average body composition, 70 kilogram male, okay? About 16-ish% of the body tissue will be made up of some form of protein, okay? That equates to about 11 kilograms, okay? This will largely be made up by muscle tissue, and that's not just skeletal muscle, i.e. biceps and everything like that, but muscle in all its forms, okay? You have smooth muscle that lines your organs. You have cardiac muscle, which is specific to the heart, okay? So you have more than one type of muscle within the body, and obviously if you're familiar with the different classifications of skeletal muscle, you know you have many different classifications of skeletal muscle itself also. So skeletal muscle has its own classifications, and then muscle itself has 3 distinct classifications also. You also have substantial amounts being present in the skin. It's roughly about 15%. There's about 43% of protein in muscle, by the way, or as a whole. 15% in the skin, and then some in the blood and in blood vessels, which is another 16%, okay? Half of this amount overall is made up of only 4 distinct proteins, that being collagen, hemoglobin, actin, and myosin, okay? Collagen is typically found in like the skin and hair. There's, you know, a lot of what's the word? A lot of beauty products and stuff like that that are targeted like skin and hair, will market themselves being like, oh my god, it has this collagen protein, okay? But that's where collagen is as well, okay? Hemoglobin is obviously in the blood, very integral to iron transport and stuff like that. And then both actin and myosin are specific to the skeletal muscles themselves, as they are what is known as contractile proteins. And basically, they are very small proteins located within the sarcomeres of skeletal muscle cells. If you think of a skeletal muscle cell as one long strand going along the length of the muscle, you know, those microscopic strands going along the length of the muscle. Sarcomeres are basically how many distinct units that is divided up into. And within those sarcomeres, you have 2 sets of proteins. You have actin, which is a small, a very, very small protein, also a small molecule. Then you have myosin, which is the large one. And how muscles contract is that those end up basically going what's called a cross-bridge cycle. Basically, the myosin pulls on the actin, almost like it's pulling on like a tug-of-war rope. And that happens all the way along in every single sarcomere over the length of the entire muscle itself. And that is how a muscle actually contracts and performs the locomotive action that is associated with skeletal muscles, okay? And that's why muscles can only pull, by the way, as well. Like all muscles, they might perform a pushing function, but the muscle itself is contracting, it's pulling closer together. And that is known as a cross-bridge cycle. That's something we can go into in another episode, probably all in and of itself of how muscles contract and stuff like that. But basically, the ends of the sarcomere will shorten towards each other, and that across the whole length of the muscle results in a contracted muscle. And that's all down to actin and myosin, pretty much, within the sarcomeres of muscle cells. That's the whole thing. We'll go into that another time. So, getting into a little bit more of the structure of amino acids and the structure of proteins now. So biochemically, the different functions that proteins are responsible for will depend on the individual sequence of amino acids within the polypeptide chain structure of the protein molecule, as well as how the protein is arranged and folded, okay? This will determine its effector location within the body, so where the protein is going to exert its effect, as well as the function that it actually does. Different amino acids have different sizes, charges, and degrees of hydrophility. Is that how you say it? Hydrophility and hydrophobicity. Okay, so propensity to be loving or not afraid of, but like averted to water, basically. Hydrophobic, you have certain ends of molecules, for example, that are what's termed hydrophobic. Think some of the little things within the cytoskeleton that maintain its rigidity against the pressure of the extracellular fluid around it in the cellular membrane. You have a hydrophobic end, which is an end that will be towards the inside of the cellular membrane, where it is averted to water, and then you'll have another end which is hydrophilictic, however you say it. Then, but anyway, that's a topic for another time. So, proteins that are built from sequences of amino acids have what is known as a side chain, basically, and this is unique to each specific protein. Okay. No two proteins will have identical side chains. The side chains vary in their atomic chemistry and structure. And this is where we're going into amino acid atomic chemistry now. Typically, amino acids will have what is known as a carboxyl group on one end of the structure. So carboxyl group. An amino group on the other end of the structure, kind of where it gets its name from. All this means is that the amino group is where the nitrogen is housed within the amino acid, okay? Typically, the nitrogen will be bonded to two hydrogens, and that's what you see if you're looking at the chemical breakdown or the elemental representation of different amino acids or whatever. You'll see an NH2, okay? That denotes the amino group, okay? And sometimes you'll also see a letter R, which is what's known as a variable group, okay? And all of this will form around what is known as a central or alpha carbon atom, okay? A term you'll be getting used to when we're talking about protein a lot is the term carbon skeleton, okay? Keep in mind that carbon is going to play an essential role when we're talking about this. So you're going to be hearing the term carbon and carbon skeleton come up a lot. Proteins sometimes also are referred to as having a primary, secondary, tertiary, and sometimes even quaternary structure. But what the primary structure is denoting is the linear sequence of amino acids held together by peptide bonds formed by chemical reaction that extracts a water molecule as it joins the amino group of one amino acid to the carboxyl group of another. All that carboxyl means, I forgot to tell you this when I mentioned it the first time, is it is a carbon atom double bonded to an oxygen atom and single bonded to what is known as hydroxyl or hydroxyl group, which is an oxygen and a hydrogen, okay? You'll often see this denoted when we're looking at atomic structures of amino acids. Again, whereas you saw NH2 to denote the amino group, you'll often see this denoted as COOH, okay? So the linear sequence of amino acids is the primary structure for the amino for the protein itself. The secondary structure is the regular folding of the peptide chain, okay? How the protein is folded. This is stabilized by hydrogen bonds between amino acids. And then finally, the tertiary structure determines the nature. I've written the nature. Determines the nature of the biochemical reactions in which the protein will engage. I wrote that down in my notes, not realizing that I've written that twice. It will determine the nature of the biochemical reactions in which the protein will engage. Okay. Some proteins, so we're going into a little bit about their functions now. Some proteins are involved in the structural maintenance at the level of the body. So looking at the skeleton, supporting tissues, connective tissues, skin and the epithelia, as I said, connective tissues and the cell. Looking at transporters, some are transporters or involved in communication throughout the body, such as plasma proteins, albumin, transferrin, and apolipoproteins. Got to try and get that all right. Apolipoproteins, hormones such as insulin, glucagon, human growth hormone, and cell membrane receptors. Some are barrier proteins and have protective functions. Look at things like keratins. You've got your skin, you've got mucin, you've got some act in the inflammatory immune response. Okay. Some are also enzymatic in nature, whether it be extracellular, such as hemoglobin. You've got oxygen and carbon carbon dioxide transport or in metabolic pathways such as glycolysis, protein synthesis, and the citric acid and urea cycles. As we'll see when we're talking about how amino acids are formed, you'll see just how much of those other metabolic pathways play a role in the synthesis and formation of amino acids that will then go on to do their distinct functions in different areas of the body. Okay, one thing you'll learn, particularly from this and any sort of study you do into human physiology, you realize how many different aspects of human physiology work together. And that what you'll see, for example, is that different proteins or different amino acids can be made from different metabolic precursors within the body from those various different systems that we talked about. It's not just dietary means that you get protein from, for example. We'll get into all that in a minute. So what happens when we eat protein then? Okay, so whether it be a steak, whether it be, I don't know, legumes, whether it be quinoa, whether it be tofu, whether it be chicken, whether it be turkey, whether it be whatever sauce, okay, you carry on down the line, list them all off, it doesn't matter. All of them pretty much digested in the same way, okay, because it all goes through your mouth, it all goes down your gullet to your stomach, and it all goes through the small intestine. So let's just take it all as we go. It doesn't matter which one you're eating, okay? When sources of dietary protein are taken into the body, they are broken down by powerful acid in the stomach. Looking at 6 molar hydrochloric acid, very, very powerful acid, and the digestive enzymes in the stomach. If you want 2 names of digestive enzymes in the stomach to wow your friends with and make them think that you really know what you're talking about, I'll give you 2 right now, pepsin and pepsinogen, okay? If you want to say, oh yeah, you know, that chicken you're just eating, oh feel that pepsin and pepsinogen, guys, yeah, if you want to try and sound smart. But that's just 2 digestive enzymes that you can list off at your next pub quiz or your next fun fact session with your friends if you want to. When they're in the stomach, they're being broken down by this acid and these digestive enzymes, pepsin and pepsinogen and others, that work to break down the bolus of protein into smaller chains of amino acids by unfolding and breaking up the large and complex mass of amino acids. And these long polypeptide chains that are arranged in this food, we want to break them down into smaller ones, okay? Much easier to digest when we break things down. The broken down bolus and the amino acid chains then move into the small intestine. So they move out of the stomach once they have been broken down sufficiently. They move into the small intestine where more enzymes, this time in the small intestine. Okay, I got 2 more names for you. So you got trypsin and chymotrypsin, if you just want 2 names of digestive enzymes that are working to break down these amino acid chains into individual amino acids. These are then taken up through the intestinal lumen via the portal vein and taken to the liver. Okay, and the liver is where the next big step of this process takes place, because the liver decides what's going to happen with these amino acids, okay? Once at the liver, and part of the amino acid pool, these amino acids can undergo one of 3 processes, okay? So number one, the liver can just use them directly for its own use, for its own needs, for its own repair and stuff like that, okay? So first off, because they go to the liver and the liver decides what's done with them, obviously the liver can decide that it needs to take priority over the amino acids that it has first, and before it sends them anywhere else, hint, hint, that's another thing it does, or uses them for any other function, it's going to make sure that the liver gets its full. Okay, liver is king, so to speak. Not liver king, liver is king. Okay. Number 2, they can then undergo the process of gluconeogenesis. Okay. What that refers to is the conversion of amino acids to glucose for energy, okay? Because free amino acids produced in this way will form the degradation of either cellular proteins or dietary proteins are deaminated to yield NH4, which is ammonia. It's a positive ammonia ion, by the way. That's all that is. And a carbon skeleton, okay? The ammonia will enter the urea cycle, and the carbon skeleton can enter metabolic pathways to generate ATP. Okay, I know I stumbled over my words a little bit there because I'm realizing I have a distinct lack of punctuation when I'm writing my notes, and it makes it rather infuriating. But basically, free amino acids can be produced when dietary proteins are deaminated, and that yields the positive ammonia ion and the carbon skeleton, whilst the ammonia ion may be gotten rid of via the urea cycle, which is a common way of how nitrogen is gotten rid of at the body as well. The carbon skeleton from those proteins, or from those amino acids rather, can be used to generate ATP via the other metabolic pathways. It can generate glucose, it can generate fatty acids, okay? Amino acids are usually responsible for about 10 to 15% of energy generated in terms of ATP yield, okay? It's a very small contribution, but it's not negligible by any means. It's not nothing. This may increase under conditions of starvation or when protein intake is massively elevated, okay? So when you've got very, very high protein intake relative to your other macronutrients, that's when you can have a much greater contribution of protein to energy than you would have if you had a much more balanced approach, okay? Finally then, number 3, they can be released into the bloodstream whereby they are free to undergo functions throughout the rest of the body. So you have the periphery tissues, such as muscle, they can go to other organs, okay? And, you know, repair the lining of other organs and all stuff like that. Okay, so those are the 3 big things that protein can do once it is broken down into amino acids through digestion in the stomach, through uptake into the liver, okay? On average, the rates of synthesis and degradation is approximately similar in adults, okay? Over time, the amount of protein in the body remains more or less constant, and nitrogen balance is therefore maintained. The key function of protein, intaking dietary protein, is that it helps maintain the balance of nitrogen in the body and helps offset losses of nitrogen through the urea cycle and through when you pee it out, basically. During growth, you have the case where synthesis exceeds breakdown and net protein deposition is achieved. You can also refer to that as a net anabolic state. I'm talking about how it contributes to muscle protein synthesis. We'll go through a little bit more about that later. Obviously, if you have the opposite scenario, the opposite happens, okay? So if you have synthesis less than breakdown, it's a net catabolic state, and over time, if that's repeated, case of starvation, for example, atrophy is going to occur, okay? Proteins will degrade at different rates. They do naturally degrade, and they degrade at different rates. So turnover times will differ significantly between different types of proteins. Some proteins degrade in minutes. There's something on my screen. It's rather annoying. Get off. Get off. Some proteins degrade in minutes, whereas others don't decay for months, okay? Very similar to like the half-life of different, what was it? Radioactive substances. That's what I was looking for. That's the word I was looking for. Radioactive substances, word of the day. No, I think the word of the day is probably going to be coming up somewhere in a minute as I try to go through some of the very complex names we're going to be going through in a minute. These are going to be words of the day. But like you have different half-lives of different radioactive compounds, you have different decay rates of different proteins, okay? Not remotely the same thing, same concept, except you have the half-life is what it takes for half of the whatever it is to decay, not the entire thing. Anyway, I don't know why I drew that comparison. As you can tell, I've had a lot of caffeine, very bouncy. So forgive me if I start going on tangents and stuff like that. Okay. To metabolize and synthesize proteins from the food we eat, a specific sequences of events at the level of the cell and the machinery contained within it has to take place, okay? 2 big words beginning with T that you're going to need to remember for this part. You first off have the process of transcription, okay? Transcription. Transcription is the process by within a nucleic cell, so that's a cell that has a nucleus, okay? By which a portion of the DNA contained with that cell, so as you know, your DNA, your genetic code, is contained within every cell of your body. A portion of that DNA corresponding to a specific protein, corresponding to a specific sequence that's going to make a protein, okay, is copied or transcribed, okay, that's where it gets its name, onto a cellular messenger, okay, onto a messenger molecule. This is known as messenger ribonucleic acid, or mRNA, okay? mRNA. Think of mRNA as the Hermes of the cell in this case, for example, or the Royal Mail, whatever service you want to describe it as, okay? But it's taken the parcel, it's taken the transcription for the amino acid to be created, and it's taken it to a different area of the cell. So yeah, this is basically a signal is sent to the nuclei of the cells of the body to begin transcribing a particular protein. And obviously it's going to depend on the location of those cells is what protein is going to be created as well. I'll explain that when we get onto an example at the end of this section in a minute. So as I said, begin transcribing a particular amino acid sequence or amino acid sequences. It's just what is needed is made clear. Next then is the process of translation. So you have transcription and then you have translation. Translation is where a cellular apparatus, if you will, or cellular organ, a little organ, or an organelle, sorry, contained within the cell, known as the ribosome, and quote unquote reads the mRNA input and encodes that portion of the DNA into a specific polypeptide chain. That is a protein. Okay. If these apparatus break down and it falls apart, we all encounter problems. Once the mRNA sequence is read and the protein is synthesized, it can then be transported to whatever site it is needed to perform its effector action. Okay, so as I give you an example. So an easy example. It's not something it's not something you'd first think of as a protein, but it is a protein because it's a hormone. Let's go through the example of insulin. See, we have more people in the world that are type 2 and that are diabetic, whether it be type 1, type 2, more predominantly type 2 nowadays. We have more people in the world that are type 2 diabetic than ever, so maybe this is quite useful and apparent for more and more people as time goes on. But let's apply this to how we would get from when we intake food to how we have insulin being produced and combating the rise in blood sugar. Let's just say we've eaten something very high in carbohydrates, okay? Let's say we've just intaken an entire pack of Sour Patch Kids. So anyway, glucose receptors in the blood vessels detect the rise in blood glucose from the carbohydrates that have just been broken down, okay? So you break down carbohydrates, you have salivary amylase and all the rest of it that's breaking down the carbohydrates, and you have a significant rise in blood glucose due to the glucose that's been created and formed from the breakdown of that carbohydrate. Okay, you had this massive rise. The signal then from these glucose receptors within the blood vessels is sent to the nuclei of the relevant cells. In this case, the beta cells within the pancreas. Okay, that's what we're saying, that different cells are going to receive different signals because they're designed for different things. Okay. The DNA within those cells is located, as I said, within the nucleus. This signal triggers the process of transcription, and the cell begins copying the relevant DNA strand that corresponds when it's translated to the hormone insulin, and that is then transcribed onto the messenger ribonucleic acid, or the mRNA. This sequence of mRNA is then read by the ribosome in the cell, encoding the polypeptide chain that is insulin, and then insulin can be released into the bloodstream to perform its vital blood glucose controlling function to maintain glucose homeostasis. As we know, when glucose homeostasis is out of whack, particularly when it's elevated chronically to the extent where it's above the well above the healthy range for humans, it leads to very bad outcomes later on down the line. And in this case, it does it by stimulating the uptake of glucose and nutrients into the muscle and into other tissues, okay? So that's how we can get from intaking food to going through those systems that we just talked about, going through those steps that we just talked about, and we end up with a hormone being created, a protein being created that then goes off to perform its function, okay? That is how pretty much all proteins are created via these processes of transcription and translation, okay? So let's look a little bit more into amino acids now. So we're looking at amino acid formation and classification. This is where you're going to get into a lot of terms you may hear a lot more personal trainers use. But when we're talking about de novo amino acid formation, de novo just means it's Latin for from the beginning, basically. That's all that means. You'll have it also using the term de novo lipogenesis, okay? De novo is just a term meaning from the beginning, okay? So when we're looking at this de novo amino acid synthesis or de novo amino acid formation, it requires that its carbon skeleton be made available from endogenous sources. And this skeleton then has an amino group added in the right position. Okay, so it's being made from endogenous precursors. Okay, give you an example. In mammalian metabolism, transaminating amino acids, such as alanine, are derived from other metabolic intermediates from the citric acid cycle. Okay, for example, that's aerobic metabolism. Okay, these can include pyruvate, alpha-ketoglutarate, and oxaloacetate. Guys, that's where I'm getting in. This is where we're getting into those complex words now. You don't necessarily need to know all of these. This is where we're getting into those complex words, which is useful because these amino acids play useful roles in, or essential roles rather, in gluconeogenesis and in amino acid transport around the body. Amino acids in some instances are also derived from other amino acids. Okay, We'll get into that a little bit now when we talk about different classifications, pardon me, of amino acids. However, the carbon skeletons, and this is hinting at what's coming next, of some amino acids cannot be attained through these means, necessitating their provision through dietary means. So let's look at some amino acid classifications. So not all amino acids are created equal. Not all of them have the same importance. Well, technically they do, because all of these amino acids we're going to be going through are just as essential as one another. That's a key term you're going to have to remember. However, the difference between them and how they're classified... Ghost? Sorry, my door just moved shut and is opening and shutting on its own. It's okay. We're not haunted, I don't think. Hopefully not. But when we're looking at how different amino acids are classified, they're not necessarily classified according to importance, because all of them are essentially important to the full optimal functioning of the body. It's just they get the different names and the different classifications due to some being able to be made endogenously in the body without dietary provision, and others are not. Some need to be provided dietarily, okay? And some need to be provided dietarily at certain times, and others then later on in the life cycle become able to be created endogenously, okay? So time is a factor, and also the nature of the amino acids themselves are a factor as well. So we're going to be classified in 2 to 3 ways, okay? You see them in 2 to 3 ways. In the guide that's available on my website, I classify them in 2. Now we're going to be talking about classifying them in 3, okay? And it's, as I said, it's depending on one particular criterion. That is, can the amino acid be derived endogenously or not, meaning within the body from what the body has or not. Those that cannot be derived from what the body already has, meaning it has to be gotten from dietary means, are referred to as essential or indispensable amino acids, okay? Those which the body can make from precursors, as we just hinted at, or through other means not requiring direct dietary intake, are referred to as non-essential, or sometimes referred to as conditionally essential, or sometimes non-conditionally indispensable or dispensable, okay? These are different terms which you just need to get to grips with. As I said, not to say that the essential amino acids or the indispensable amino acids are more important than the dispensable or non-essential amino acids. It's just that how they are gotten is different, and one needs to be provided through food, whereas the other doesn't. There are 9 essential or indispensable amino acids, and there are 11 that are deemed conditionally essential or conditionally indispensable or non-essential and dispensable. The essential amino acids, so let's list them off. Leucine, isoleucine, valine, histidine, threonine, phenylalanine, lysine, methionine, tryptophan. Those are your 9 essential or indispensable amino acids that have to be provided through dietary means, okay? Leucine, isoleucine, valine, tryptophan, lysine, methionine, phenylalanine, threonine, histidine. Obviously, they play various roles within the body. Some of them are more specific to muscle tissue, skeletal muscle tissue. Some are involved in other things like amino acid transport. I just realized the dog has led down. Bailey's led down, and she's led right down underneath her water bowl, which is quite a large bowl, and she's tipped part of the water all over the carpet. Just saying, Belle, Mom is not going to be happy with you. But then again, it's in my room, so maybe she won't care. So you have conditionally essential or conditionally indispensable amino acids. You have arginine, cysteine, glutamine, glycine, proline, serine, and tyrosine. Okay? There's 8? Yes. Oh, no. Oh, I added something up wrong. Arginine, cysteine, glutamine, glycine, proline, serine, tyrosine. 7. Yes, 7. Finally, then you have the non-essential ones, which is alanine, asparagine. You have glutamate, also sometimes referred to as glutamic acid. And then you have aspartate, also sometimes referred to as aspartic acid. Okay, alanine, asparagine, glutamate / glutamic acid, aspartate / aspartic acid. Okay, so those are, if my math is correct, those should add up to 20. I hope. I hope. So the reason why some are called conditionally essential, so let's go through this first, is due to them being amino acids that under certain physiological circumstances, they become essential, okay? Due to the endogenous production not being sufficient to meet the demand, or during under certain disease states or maturity states, okay? For example, conditionally essential amino acids can become essential under certain disease conditions, okay, like I said, where the endogenous production of them is negatively impacted by the disease state. Another example is during childhood, where a number of amino acids that are not essential for adults are essential for children, as the amount required cannot be met by endogenous production only yet, because this is due to those pathways, the body's pathways for formation of these amino acids not being fully matured to the extent where they can support the growth of the child on their own, okay? So you have some that become non-essential later on in life simply because the body's more mature and it's able to be more self-sufficient, okay? The metabolic pathways have matured. And then you also have some that when you're under certain disease states, they actually become essential, okay? So you have some amino acids that start off as essential and become non-essential, again, not meaning they're less important. But then you also have some that start off as essential and become non-essential. Okay, but depending on where you are in your life cycle, that's going to be obviously important to know. If you've got children, for example, it's going to be important to know. So here we go. These are where I'm going to give you an example here of how different amino acids can be synthesized from different amino acids. And also from the other energy systems within the body. Get ready for me to probably struggle to pronounce some of these very much so too many syllabled words, as well as numbers, like not too many syllabled numbers, but numbers within the words that have too many syllables. And yeah, so when we're looking at a lot of these amino acids that can be synthesized from intermediates of the body's energy generation system, such as the process of glycolysis and the TCA cycle, these are just other metabolic pathways by which ATP is generated, and ATP is the body's energy currency that is responsible for creating the energy that produces all of the actions at the scale of the cell within the body. It's responsible for every life-sustaining process. As I said, and then if the other amino acids are available in sufficient amount, they can be created from those also. An example of this is what happens with the glycolytic intermediate 3-phosphoglycerate, okay? This undergoes several processes and generates serine and glycine. So as you remember from what we just went through, serine and glycine are two conditionally essential, conditionally indispensable amino acids, okay? They can be derived from this intermediate of glycolysis, 3-phosphoglycerate. 3-phosphoglycerate also gets converted into pyruvate, okay? This is then fed into the TCA cycle, okay? The formation of pyruvate is a critical step within the citric acid cycle or the tricarboxylic acid cycle, okay? But it also undergoes conversion into 3-phosphohydroxypyruvate via oxidation, courtesy of the 3-phosphoglycerate dehydrogenase enzyme. Typically what you'll find, anything that ends in ase, i.e. ASE, will typically be referring to an enzyme of some kind, okay? And then the reduction of NAD plus to NADH as well. 3-phosphohydroxypyruvate then undergoes a glutamate-linked transamination reaction, okay? Called, or is it catalyzed by phosphoserine aminotransferase 1, and this forms another intermediate known as 3-phosphoserine, okay? Typically what you find is that the end name of the intermediate or the molecule will contain a good part of the enzyme that created it. Okay, that's typically where it gets its name from. This reaction also results in glutamate becoming alpha-ketoglutarate. That is also then fed back into the TCA cycle. Finally, this is then hydrolyzed by the phosphoserine phosphatase enzyme to yield serine itself. Okay, so then yields the amino acid serine. Okay, so starting all the way back at 3-phosphoglycerate, you've gone through all of these processes, you end up with serine. Okay, you can also get glycine back from serine then. The enzyme, oh, here we go. Serine hydroxymethyltransferase. Serine hydroxymethyltransferase facilitates the reaction whilst also coupling it to the reaction of tetrahydrofolate, or THF. Coupling it to the reaction of tetrahydrofolate to N510 methylene tetrahydrofolate. Okay, so yeah, a lot of big words. I told you these are where you're going to be. These are where your words of the day going to be coming in. Serine is also required for cysteine production. Cystathione beta synthase converts another compound known as homocysteine and serine into cystathione, and cystathione gammalase converts this into cysteine. So that's a lot of big names, but that, for example, I think it might have a little diagram in one of the studies I'm going to be linking down below to illustrate where or how this all links together in like a graphical format. Not exactly the easiest thing to understand, but this is just where by all of these different enzymatic and hydrolysis reactions, you can get different things. And this is stuff that the body just does like that. This is how the body is an absolutely amazing machine, okay? It just does this shit. I gotta try and like pronounce all this stuff and get it across in a way that's easy for you to understand. Because if I'm fucking up the pronunciation of things, obviously it's going to be hard for you guys to understand. It's going to be hard for you guys to remember. Not that you necessarily need to remember it, but it's like this is just stuff the body just does 24/7, 365 from conception to death, near enough. You know, this is just like, obviously the body doesn't necessarily know that it just is. The body just recognizes it as an atomic elemental structure. And then we come up with all these weird, stupid fucking names for them. But I'm just like, you know, it's absolutely insane that all of this stuff is just going on at the atomic level. And we don't have no idea. We have absolutely no idea. And the body's just going, just ticking it all over. It's absolutely insane. It's absolutely insane. Anyway, the conditional and non-essential amino acids also play roles then in synthesizing other important compounds within the body. Here we go. I'm going to give you a few examples. So glutamate, for example, acts as a nitrogen donor and an acceptor, and is a central amino acid for the movement of nitrogen among amino acids. Okay. Then you also have tyrosine. We mentioned that before. Is the precursor to norepinephrine, epinephrine, dopamine, and melanins. Tyrosine itself is derived from another amino acid known as phenylalanine. Okay, so you have the essential amino acid phenylalanine. Try pronouncing that 5 times fast. And then that itself allows tyrosine to be derived from it. Okay, and this is in the absence of and in addition to dietary consumption. Then you have methionine, provides the methyl group for many DNA and histone methyltransferases to regulate epigenetics. You have cysteine, glutamate, and glycine generate the antioxidant glutathione. So you have nitric oxygen synthases as well, use arginine to generate nitric oxide. This is a potent muscle relaxant, and it is derived from and biosynthesized from the terminal guanidine nitrogen atom of arginine, okay? Its generation also produces citrulline, okay? Citrulline is a common additive to pre-workouts due to its effects of also relaxing blood vessels, causing them to expand, vasodilation. Nitric oxide, by the way, or citrulline rather, is very good or very important for its roles in controlling tone of your blood vessels, okay? So blood vessels, as you know, your arteries, your veins, they have the capacity to expand or dilate, as known as vaso meaning vessel, dilation meaning increase in size, basically, expand, and then you have constrict, so you have vaso. Oh no. I've lost focus on the other camera. Why is the other camera lost focus? Yeah, I think I've reset it. Okay. Oh shit, I lost where I was. I said, yeah, it allows the blood vessels to dilate and it allows to constrict. Okay, and citrulline is very important for monitoring this tonality. Okay, or regulating this tonality. I said when it allows for increased vasodilation, for example, when you're training and you want to get a big pump, increases blood flow and nutrient flux into the muscle, which aids in improving performance, particularly regarding muscular endurance under fatigue. Okay, so this is very, very important. Also, glycine and glutamate also serve as neurotransmitters. Glutamate can also generate another amino acid transmitter, gamma aminobutyric acid, or GABA. Then you have glycine and glutamate. They also serve as neurotransmitters, okay? And then also tryptophan is used as the precursor for the generation of the neurotransmitter serotonin, okay? Serotonin plays vital roles in, well-being and feelings of dominance and stuff like that. Serotonin is also used to generate melatonin, which helps regulate our sleep cycles and our circadian rhythms, okay? So all these very, very important. All these very, very important. So there's a good example of how conditionally essential amino acids and non-essential amino acids also play important roles, and how they are synthesized not only from metabolic precursors but also other amino acids also. We're going to touch on something I mentioned earlier, which is something that provides an important distinction with protein versus any of the other macronutrients really that are stored, okay? This is what is meant by the amino acid pool, okay? I mentioned it earlier when we talked about amino acids being uptaken into the liver and into the amino acid pool. This is just a term to denote that there is no consistent, unchanging reserve, really, of amino acids. Unlike body fat, which is like just a massive energy reservoir that doesn't really change much over time, unless put under conditions of energy deficit, in which case it will be lost over time, for example. But the level of amino acids and circulating amino acids within the body changes much more rapidly and acutely relative to body fat. Amino acids are constantly being put into and taken out of the amino acid pool. And this is coincided with the cycles of anabolism or synthesis and catabolism or breakdown, okay? And it's why they need to be quite regularly replenished, and a good amount of them need to be taken in order to offset things like nitrogen loss and stuff like that. How do we measure it, though? So the amount of protein within the body cannot be determined directly via measurement, obviously, because it is constantly changing. However, one way in which protein status is estimated is in a laboratory setting. Okay, this is just something for your general knowledge and stuff like that of how, protein status within the body is measured. And if this comes from measuring the nitrogen content of the body. So you have nitrogen balance, as most of the nitrogen contained within the body is present as amino acids, okay, in protein. I said all amino acids have that amino group, which is NH2, where the nitrogen is, okay? And most protein is present as lean tissue, okay? Scientists can then use a procedure known as in vivo neutron activation analysis to determine the nitrogen content of the body, and that will give an indication of protein then, or amino acid. On the other hand, indirect measures of estimated lean body mass, such as bioelectrical impedance, so that'd be like an in-body, which we talked about a couple of podcasts ago. Hydrodensitometry, for example, can be used to estimate lean body mass and other physiological parameters, such as fat mass, for example, on the assumption that protein content of the tissues remains relatively constant. Okay, so it may remain relatively constant over time, but the issue, it is constantly changing. It's constantly changing. So the amino acid pool, if you ever hear this used, is just a term used to describe the constant cycle that is ever occurring in all of us 24/7, 365, where amino acids are added to this pool, not only from dietary sources, obviously from absorption, but also through recycling and repurposing of old proteins that have been partially degraded and just need repairing or replacing. As I said, the cycle is constant. You're constantly going through, and this is where we're talking about muscle protein synthesis. I like the way that Lane Norton describes it, for example, where he says protein synthesis is just like a dimmer switch. It's constantly happening, and the light is never fully on, but it's never fully off either. So even if you're in a state of like very high muscle protein synthesis, I think it might be this way around. I don't know which way around it's going to be. I want this to be on the right-hand side if possible, but I think it's going to be. Either way, when you're going, when you're just taking in a bunch of protein, for example, you just had a massive meal and you're very high in terms of muscle protein synthesis, it's never fully on because you still got a little bit of breakdown occurring. One's never fully off, one's never fully on, okay? There's also the continuous breakdown of protein that results in energy being produced, as I said, when carbon skeletons of the amino acid molecules oxidize to form water and CO2. Okay, and then the amino group, where the nitrogen is, of the protein is then fed into the urea pool, and then that urea will be excreted. Okay, so that's a lot on the background of protein. We've done just a general bit about how proteins are formed from DNA and our genetic code. We've done, what happens when we eat it, how proteins are digested, all the way, and then how different amino acids serve different functions, how each function is determined, or how they're going to do what is determined. The amino acid formation itself, the classification, amino acid pool. There's a lot of things that we've done. I swear to God, this house is haunted. A lot of things we've done so far. I'm just going to go through a little bit now, which is going to be more useful to you guys if you have interests in, muscle hypertrophy, strength, for example. I've titled this section of it Building and Breaking Muscles, but it's going to be more specific to skeletal muscle protein synthesis now. And we're going to be talking specifically about skeletal muscle tissue. As I said, muscle tissue is constantly going through cycles of synthesis, which is also known as anabolism, and breakdown, or catabolism, okay? As I said, literally just said this about 120 seconds ago. Process is very much like a metabolic dimmer switch, okay? One process is never fully off and the other process never fully on. So anybody says about switching on protein synthesis, or you can't do this because you're going to switch off your ability to build muscle, never works, okay? Never works, okay? Just disregard them completely. That's like a V-shred thing to say. One is never fully off, the other's never fully on. You're constantly going through cycles of catabolism and anabolism. You should be anyway if your protein intake is good. Under certain changing circumstances, however, the balance between the two will shift heavily from one to the other. Okay, so you could skew the balance heavily from one to the other depending on what you do. And if you skew it heavily in favor of growth, you're going to grow over time. If you skew it heavily in favor of breakdown, you're going to break down over time, naturally. So the end result is going to depend on what side of the scale or how the balance is over long periods of time on average, okay? This is what sometimes you refer to as net anabolic state. Okay, I know James from Shredded Sports Science uses that term a lot. It's like the average, basically. You will go through phases of breakdown inevitably, okay? Sleep is the biggest one, okay? You need sleep to fully recover, but sleep is one of the times when you're going to be breaking down because you run out of, available amino acids, free amino acids midway through the evening, and you end up in a catabolic state by the morning, okay? But you're going to constantly be going through these series of synthesis and breakdown. It just matters which one you're in more. It matters which one you're in more than the other. Okay, that's what really matters. It's impossible to not be in states of breakdown. In fact, it might be counterproductive as well. Okay, let's look at these examples then. So let's just say you just finish a massive Sunday roast, full of meat, starchy carbs, gravy, the lot, okay? After finishing it in an abundance of energy, protein, other nutrients, okay? Body's in prime condition to begin the process of synthesis. And that's provided you give it a stimulus for growth as well. So that's your resistance training, that's your exercise. But provided it has a stimulus for growth, and that'll massively upregulate the synthesis, the muscle protein synthesis itself. And that'll provide, as I said, further stimulus for growth. But when you eat, you're in prime condition for muscle protein synthesis. And that's when it's going to be highest. Just after you've eaten is when it's going to be highest. Whilst the small amount of the overall protein you eat will go towards building skeletal muscle, being in abundance of energy with a lasting stimulus for growth ensures a maximal response, okay? Dimmer is set to very bright in favor of synthesis. That's another big thing as well. You'd be surprised how little the protein you intake actually goes towards your muscles by the time there is enough left over after it's completed all its other essential functions for muscle protein synthesis. And that's why as well you have to eat so much protein in order to maximize. That's why you have to eat so much protein relative to your body weight, for example, to maximize muscle protein synthesis, because you have to get over this hump first of protein having to fulfill other essential functions and then making sure you have enough left over in order for it to be diverted towards muscle. That's why of the overall amount of protein you eat, it could be as little as, for example, 10 grams of that per day will be deposited in muscle tissue. But you extrapolate that over a year, That's a lot. That's quite a bit. But in order for you to set the dimmer to very, very bright, you have to input a lot of protein into the system, basically, okay? That's why big misconceptions around you can only digest a certain amount of protein and all stuff like that. Massive misconception. But let's just say then, second example, you wake up in the morning after a long and restful 8 hours. I know it's an imagined luxury for most, including myself, but yes, anyway. Last meal was 8 o'clock in the evening. Let's just say that you went to bed at 11 p.m. You wake up at 7, stay in your bed and your phone for an hour. So 8 o'clock at night to 8 o'clock in the morning. Okay, 12 hours since you last had any food, let alone protein itself. Currently, the switch is set to very dim, okay, in favor of breakdown. During the night, you may have been resting, but obviously body was not. It was still going through the amino acids it had available. And breakdown will temporarily exceed synthesis. When you eat, then the balance will skew back in favor of synthesis again, okay? Until the digestion finishes, and then you'll gradually start making your way back down towards catabolism again. But these cycles of anabolism and catabolism are very necessary, very, very necessary and unavoidable. So, without anabolism, you have little to no growth. Without catabolism or breaks in the anabolic processes, there's no recycling of old and degraded proteins, and the anabolic response has the potential to become blunted over time. That's why you need to have breaks in between your meals or servings of protein, okay? Doesn't just turn off and on. And you need to give yourself breaks in order for the dimmer switch to start going back towards breakdown, in order to resynthesize yourself or resensitize yourself to protein intake, so that your body doesn't basically just get exhausted by it. Okay. What are the big things that you need to keep in mind or prioritize when you're looking at protein to maximize muscle protein synthesis? First one, overall protein amount. Let's go into focus again. I can see it. First one is overall protein amount. Second, protein quality. And third, meal frequency. I'll put like protein quality and overall protein amount, I put as a tied first, and then meal frequency I sort of put as a close second. But really, all of these are like things you need to do well. They are all like equally things you need to do well. The number, if you had to pick number one though, probably would be overall protein amount. Absolutely. So overall protein amount, I've said here, is the single most important factor for maximizing muscle protein synthesis in the pursuit of maximizing skeletal muscle hypertrophy and getting the most muscle that you can. As I said, you'll see how this fits into the picture when we talk about the other two in a minute. So up to a point, maximal muscle hypertrophy or maximal muscle protein synthesis increases in drastic fashion with increased protein uptake, up to about 1.6 grams per kilogram of body mass per day. Upper thresholds of what has been deemed to be effective are suggested limited return on investment, if you will. I'm just going to be using the other camera, I can tell. I'm just going to be using the camera in front of me at this rate, because I don't know how often that's going out of focus. So, up to a suggested limit, of 2.2 to 2.5 grams per kilograms per day, if you really want to maximize muscle protein synthesis, okay, and muscleize your chance of getting... did I say muscleize? Did I just say muscleize? I think I did. You maximize your muscle hypertrophy output, okay? In this range is the sweet spot where most, in terms of protein intake, will lie, okay? Many don't even hit the bare. This is the thing you'll learn when you first start recording your protein intake. I don't know whether it's just using, protein reference ranges from Google or, on MyFitnessPal or any other service, okay? You can use mine if you want. Many don't even hit the bare minimum for sedentary individuals, okay? And the bare minimum for sedentary individuals is 0.8 grams per kilogram per day, okay? You think about it, let's just take a 100-kilogram individual, not doing any exercise, working in an office all day, sits in front of the TV watching Netflix for hours a day. They don't really do anything. Steps less than like 3,000 a day. Just going back and forth the fridge for their daily activity, basically, aside from going to work and commuting in the car or the train or whatever it is. That would be 80 grams per day. That would be 80 grams per day. Just easy to do it off 100 kilos because obviously it's just 80%. So yeah, that's like a bare minimum for them. Obviously, if you have less body weight, it's slightly less, you adjust it. But many people don't even hit this. I've done, look at people's diets and they're getting in like less than 50 grams of protein a day. And I'm like, how the hell are you surviving? How the hell are you surviving? You don't stand a chance of gaining any muscle or stimulating muscle protein synthesis with intakes this low. Okay, you do not stand a chance. Don't matter how many times you go to the gym. Well, you can go to the gym, it will improve it a little bit, but you don't stand a chance gaining appreciable muscle with this. The little intake will be solely used within the liver and for energy generation, and you won't have anywhere near enough left over. You won't have anywhere near enough left over to gain appreciable muscle from it. So you need to be intaking more. You need much more. So overall protein amount is one of the three kings. We'll call it the three kings. I think, wasn't there? Oh no, it was the four kings, wasn't it? There was a boss battle in the original Dark Souls. There was a boss battle in the original Dark Souls game. There was the four kings, wasn't it? It was the four kings in the Abyss. I never got to that section of the game. I was never good enough, but I know it existed because I looked up the ending to the games and all stuff like that. But yeah, it's like these are like the three kings. You got the four kings in Dark Souls. These are the three kings of maximizing protein and energy and muscle protein synthesis, if you will. Protein quality refers to the type of protein you get from, and going off what we've discussed regarding essential amino acids and amino acids, it's very important to consider. This refers to protein quality, comes in two branching terms, and you have complete or incomplete. You have complete or incomplete proteins. That's how they're referred to, okay? What gives them this name is if they have all 9 of the indispensable or essential amino acids present within the food matrix that you're eating in sufficient amount, okay? If they do, if they have all 9 essential amino acids in sufficient amounts to stimulate protein synthesis fully, that is referred to as a complete protein, okay? If they do not, they're classified as incomplete because they have 1 or more amino acids either missing in sufficient amount or missing in presence overall, okay? Animal proteins are complete proteins, okay? So animal proteins, for whatever reason, you know, it happens to be this way, is that they contain all of the essential amino acids in different amounts, but all of them in sufficient amounts for the serving sizes that we eat as humans, okay? So there's no guesswork with animal proteins, they all have them, okay? Plant proteins, on the other hand, are not the same, okay? Some of them, quite a few of them, tend to be lacking in 1 or more of the amino acids needed for muscle protein synthesis or protein synthesis to be stimulated fully, okay? Typically, lysine and methionine are the 2 that are missing, either in amount or in presence overall. You do have complete plant proteins, tofu, quinoa. They are complete proteins, but many tend to lack 1 or more of the essential amino acids, meaning that the deficit from 1, you can account for it because the deficit from 1 just has to be made up by a source of that amino acid from another food source, okay? So that's why what you'll tend to find is people who want to eat more of a plant-based diet, they can still get sufficient protein, sufficient amount, and sufficient amino acid, you know, amount, and all of their essential amino acids. They can do it. It just tends to be a little bit harder, especially if you don't know what you're doing. It can be a lot more difficult, okay? Whereas animal proteins, they have all of them. There's no guesswork. You don't need to worry about it, okay? Bioavailability also plays a role in protein quality, okay? Bioavailability refers to how much of a nutrient contained within a food matrix is able to be accessed and absorbed via digestion, okay? Foods with high bioavailability means that the vast majority of the nutrients contained within the food will be assimilated and absorbed. That's having a high bioavailability. This will mean that less of the nutrients, if a food has low availability, low bioavailability, less of the nutrients will be uptaken and more will be lost, okay? Typically, this is due to differences in the structure of the food matrix when you get down to the level of the cell. Think about it, whether you're eating plants or whether you're eating animal products, they're all cells. They're all cells, okay? You're eating animal cells when you eat a steak. You're eating cow cells, okay? Similarly, you're eating plant cells when you eat plant-based foods, okay? Animal sources tend to be more generally bioavailable as the structure of their cells are relatively similar to ours. Namely, they lack a thick protective cell wall on the outside of them. We only have a cell membrane. Animal cells only have a cell membrane, okay? digestion, assimilation, and absorption, therefore, is much easier. It's easier to break this down because we don't have a massive wall that's thick and hard to break down around the outside, okay? Plants, on the other hand, with their thick protective walls, again, this is thicker on a cellular scale, okay? So, you wouldn't be able to see it, but relative to the size of a cell, it's very much so, drastically increase in size and thickness. It makes breaking them down much harder, possibly impossible, as we lack the digestive machinery to fully break them down and digest them, okay? However, this is why we tend to say that a lot of plant-based foods are high in fibers, because they can't be digested, is then what allows them to exert positive functions within the gut itself, and it helps clean them out. A lot of things a lot of plant foods are deemed high in fiber because they can't be digested, okay? You have insoluble fiber, for example. So, this is a key example, okay? There's a reason why we can't survive, for example, on a diet of grass alone, but a cow can, okay? This is where you have to take into account that different organisms are very, very different. As well, people will think like, oh, look at cows, they're massive, muscular, hulking machines, and all they eat is grass, okay? But you have to take into account their digestive machinery is a lot different to ours, and it's a lot more suited to breaking down plant chemical structure. Cows, being ruminants, for example, have much larger digestive machinery. Their stomach, and their stomach in particular, has 4 distinct compartments, okay? And they are able to break down and digest plants on a scale, or to an extent, that our measly one stomach, even with its 6 molar hydrochloric acid, could never aim to replicate. And that's not what we're here. That's not what we're here for anyway. That's not what we're here for. We're not here to eat grass. We're not grazers. Our digestive machinery could never do the same thing that a cow's digestive machinery can do. However, we can get a lot of good nutritious value and taste from cows because they not only taste great. May or may not have offended a bunch of vegans in saying that. They not only taste great, but the cells that make up their biology are much easier for us to digest. Cells that contain within their muscle and stuff like that are a lot easier for us to digest, lacking that pesky cell wall. And so we get a lot of nutritious value from them because they're more bioavailable. So you must take caution with bioavailability also. And that sources of nutrients that are less bioavailable translate to less of their nutrients coming across to us during digestion, and that may also need to be accounted for. So that's protein quality. You want to make sure that you're having complete, high bioavailability sources at every single meal if you can. Animal products, whey protein supplementation. You can eat plant proteins, obviously. That's no problem at all. You can get everything you need on a plant-based diet. Nobody's saying that you can't. Nobody's saying that you can't. It might just be harder. And obviously you want to make sure that those sources that you're taking in, that they are as bioavailable as you can get. Okay? Last one then is meal frequency. So meal frequency is very often misconstrued. And this was one of the things that really got me down when I first learned about nutrition and wanting to improve my physical appearance and all stuff like that. That's a big reason why I wanted to become a personal trainer, for example. It's often misconstrued aspect and factor, especially regarding protein. You got the people that say for every person rather, that gives the gotta eat 6 meals a day to maximize anabolism, brah. There's 1 meal a day is all you need, guys, as well. You know, for every person that says you gotta skip breakfast, there's a person that says you gotta have 10 million protein shakes after you train because your anabolic window is 30 nanoseconds long. So how do we make sense of it? How do we make sense of it? Well, meal frequency really just depends on the context. As previously discussed, matters even really more so is the overall amount of protein, okay? So if you're not eating enough protein, doesn't matter whether you eat that small midget of protein over 5 meals or over 1, it doesn't really matter. In fact, the 1 meal is probably going to be better in that instance. Meal frequency is important, and there is a definitive frequency range that gives you the best return on your investment, So it appears to be about 3 to 5 times per day, okay? Provided the serving sizes and your protein intake is sufficiently spread out across those servings, okay? Muscle protein synthesis is a curve, okay? So it spikes initially after you intake, and over time it gradually comes back down. Okay. Now, different proteins provide different protein synthesis curves. Okay, because some proteins, they spike your protein synthesis very quickly and then come down very quickly. So that'd be something like a whey protein, for example. Whey protein provides a massive stimulus for muscle protein synthesis, but it doesn't last as long. Contrast that with casein, for example. Casein protein doesn't stimulate muscle protein synthesis to the same degree in the short term as whey does, but it lasts a lot longer. The curve is flatter, but it's longer. Okay, so that's why, like casein, for example, a lot of people recommend that you take it before you go to bed, so that the catabolic impact overnight isn't as bad. Okay, because it takes longer to digest. Okay, that's why when you mix it in with things like protein shakes and maybe protein porridge and stuff like that, it actually like is a lot harder to blend in because it's a lot thicker. Okay, but then the 3 to 5 times per day, provided it's spaced out evenly and with sufficient time in between them. Okay, you need to wait for that curve to start coming back down before you eat protein again. That's what we meant by, if you try and keep that curve all the way up, all the way up, all the way up, you end up getting a massive diminished return, because you need to wait for the mechanism of protein synthesis to basically resensitize before you take your next serving size in. You need to give yourself time where you're not eating protein, and where you're not eating anyway. You need to give yourself that time, several hours between feeding, so that enough time elapses, and digestion is going to be in the range of about 3 to 4 hours this time frame, by the way, maybe a little bit longer. Absorption and utilization is allowed to proceed unimpeded. Okay, depending on what your life schedule is like, the optimal frequency will vary. Okay, for most, it will likely be 3 to 4. Okay, depending on obviously when you wake up as well. If you wake up very in the morning and then go to bed and you're like a protein shaker right before you go to bed at night, you can maybe squeeze 5 in. But then for every one you add sort of above that point, if you try to guess, 6, 7, 8, 9, you're getting such a diminished return on investment, if you're getting anything. Okay, above 5, that's like the top of the top. Okay, if you're awake long enough. Okay, shouldn't necessarily sacrifice sleep either to get more in. It's just that if you wake up, for example, say you get 8 hours, for example, let's just say you go to bed at 10, you wake up at 6, you had a protein shake right before you went to bed about 9:30, and then you have, again, maybe another protein shake when you wake up, or you have a meal straight away when you get up at, 6 in the morning. Then obviously, then you get 10 o'clock, 4 hours later, you get 2 o'clock, that's another 4 hours later. You get 6 o'clock, that's another 4 hours later. So that's 6, 10, 2, 6, and then one before you go to bed again. You can get 5 in very evenly spaced out that way. But then for some people, they might, naturally just sleep a little bit longer or have a little bit less time throughout the day. Maybe they get 4 in, maybe they get 3 in. Okay, at that point, what matters is that you're just getting enough in every single serving. So your protein serving is going to be bigger if you're eating less meals throughout the day. Okay, so that is basically the ins and outs with regards to meal frequency. Okay, 1 and 2 is very likely. 1 is going to be very much suboptimal. 2 is going to be suboptimal, at least for maximizing muscle hypertrophy and strength. Then if you can get 3, between 3 and 5, that's going to be the sweet spot for most people. And that is an hour and 15 minutes of me talking about protein. I think we're going to leave it there for today. But no doubt this will help you guys out a fair bit with your background understanding of protein, and the how proteins are formed, how they're useful, why you need to eat a certain amount if you've got a certain goal, or the other things that protein takes into account. Obviously balancing nitrogen, not just aiding in muscle hypertrophy, balancing things like nitrogen and all stuff like that. Yeah, that's gonna be it from me today. We can do this for some of the other macronutrients as well. Give me a bit of time. But I hope you enjoyed that, guys. I hope you find it useful more than anything. Hope you found it educational. And yeah, hopefully it gave you some useful things to work with as well, so that if you've got a goal with regarding muscle hypertrophy, or you want to help people that understand why protein is important and how it's digested, and help them understand some of the misconceptions they might have about nutrition. Going to be useful for you, obviously, in that regard as well. So, as I said, I hope you found this episode useful, guys. I hope you enjoyed it. Hope you enjoyed it. hope you're still in focus. And from me, and from Bailey, who's currently sleeping down there, but from me, and from Cushing Bailey, we hope you've enjoyed it, and we can't wait to see you again soon. Hope you have an amazing day, guys, wherever you are. Amazing day, morning, afternoon, evening, night. And yes, if you'd like some help with your nutrition or like training, then maybe useful checking out some things that we've got down below. Thank you, guys. I'll see you again soon. Out.